Good afternoon, everyone. Welcome to this installment of the CORBEL webinar series. Today, we are very pleased to have with us Christian Tischer from the Centre for Bioimage Analysis, EMBL, to present a basic introduction to bioimage analysis. My name is Michelle Mendonça, and I'm involved in the CORBEL project on behalf of EMBL EBI, and I will be hosting the webinar today. I'll also be manning the question function. Before I introduce our speaker, I want to make you aware that this webinar is being recorded, including the question and answer session at the end. It will be made available on the CORBEL YouTube channel and the website. Now, we have reserved some time at the end of the session for questions and discussion. I would like to ask you to write your questions in the question function of the GoToWebinar control panel, as you can see on the slide. Now, I would like to briefly introduce the CORBEL project to you. CORBEL is a Horizon 2020 funded project, bringing together 13 research infrastructures in biomedical science. CORBEL aims to transform understanding of biological mechanisms and help translate them into medical care. Modern biological and biomedical research involves complex projects which often combine a variety of different technologies and operate at the interface between different disciplines. CORBEL aims to help these projects by harmonizing access and services for research involving more than one research infrastructure, which could be biological and medical technologies, biological samples, and data services. Our presenter today is Christian Tischer. Christian studied physics in Heidelberg, followed by PhD at EMBL with Philippe Bastiens, working on microscope development and signaling in mammalian cells. Subsequently, he did a postdoc with Marleen Dogetom at Omolf in the Netherlands, working on microtubule dynamics. From 2009 until 2018, he worked at EMBL's Advanced Light Microscopy facility, supporting scientists with both microscopy and image analysis. Since the end of 2018, Christian's focus shifted completely towards image analysis support, and he is now running EMBL Centre for Bioimage Analysis. At this point, I will hand over to Christian.
Very good, thank you. So my name is Christian, as has been mentioned, and I want to give a short introduction to bioimage analysis. So what is bioimage analysis? Actually, it's a kind of new thing. So one could say it's a developing field in computer science, and I tried in the, oops, I cannot go forward. In the next slide to give a kind of definition, which is always hard, but I think one could say bioimage analysis is about to automatically extract biophysically meaningful information from microscopy images of biological samples. And I colored all these words in different colors to just actually highlight that it's a quite complex discipline, because automatically implies that the person that does that has some kind of computer science background, so is able to write code. The extraction process is actually really like the core thing that you have to know, like how to devise image analysis procedures and algorithms to get information from images. But the challenge comes that it shouldn't be just some image processing, like it's maybe done in Photoshop, but it has to be something that is biophysically meaningful. So the person that does that also has to understand the biology behind the project. Then it comes from microscopy images, and as you may know, there are very different modalities of microscopy, and almost every year we get a new one now. So there's electron microscopy, confocal microscopy, wide field microscopy, super resolution microscopy, light sheet microscopy. In fact, all of these images have to be treated a bit differently, so that actually one almost has to specialize already as a bioimage analyst. And last but not least, these are biological samples. So to properly do the job, you also really have to have a good deal of knowledge about biology. So since all of this is actually quite demanding, there is now a new profession emerging of people that are just doing this, and they do it in a high quality. And these people call themselves now bioimage analysts, and I would call myself like that as well. Also, the EU actually recognized that there's a need to have professional bioimage analysts, and that's why there was or is a EU-funded cost action namely the network of European bioimage analysts. And apart from giving courses and organizing conferences, another main focus of this action is actually to establish the point that there should be a profession of bioimage analysts. Okay, how does bioimage analysis help you? If you look at these two images on the left, you may see that there's normal cells, they have normal nuclei, whereas here, the treated one, the nuclei, most of them look a bit different. In fact, they are stuck in mitosis. Now, maybe 10 or 20 years ago, it was still state of the art if you want to publish your finding that the treatment does this change, to publish these two images and say in the figure legend that they are representative. I think now you probably will not be able to do that, but the journals will ask you for quantification. And that's basically where you need bioimage analysis. So you should first of all quantify this. So for example, compute what is really the fraction of non-interface cells, so quantification. Then this should be reproducible. And that typically means that you shouldn't necessarily do this manually, but devise some computer program that does that so that other people can reproduce your analysis. And you need statistics, which means you shouldn't do it only on 2 images, but on many. And again, that requires automation. So that's basically 3 of the main things: quantification, reproducibility, and statistics. And this is now kind of required for most publications. Okay, let's dive in to understand in a bit more detail what bioimage analysis is. We have to think for a moment what an image actually is. And my attempt here is to say it's a bunch of pixels. And a pixel has basically 2 main properties. One is a value, and the second thing, it has a coordinate. So it lives somewhere in space. So if you look here, this is in fact a 2D image. So we have an X coordinate and a Y coordinate, and at different coordinates we have values. And this is also how computers like to look at images, but for a human brain, this is not very digestible. That's why we typically do a trick, and we apply a so-called lookup table, which means that we assign numbers to colors, and then we paint these colors at the respective coordinates on the screen. And then this is much easier to see what this image actually contains, namely some nuclei. So here a grayscale lookup table has been used. This is not the only thing you can do. Actually, another quite popular choice is these false color lookup tables. And that, for example, as you can appreciate here, helps you to also see the dark halo around the nuclei better, which is not visible in the grayscale lookup table. So I think there basically everything is possible. The point is just it's best practice to show in your images this scale bar here, so that people can really understand which number belongs to which color. Something that's not recommended is what comes up now, and this is basically using red lookup table on black, because I personally, for example, don't have a red weakness, and I think 15% of our viewers now see nothing on this upper image, and that's still a common mistake. So if you give a talk, for example, with this image, you will lose a good fraction of your audience because they just see a black image. So basically the red lookup tables are maybe the ones that you should avoid if possible. Okay, another very important technical thing about images is that they also have a data type. And so basically these values of the pixels, they are limited. They cannot take all values. So for example, you might have heard of 8-bit images, or it's also called unsigned byte images. So they can only take integer values starting from 0, 1, 2, 3, and then the maximum value 255. Which is actually 2 to the power of 8 minus 1, and that's where the 8-bit comes in. Now, if you work with such data, you have to kind of appreciate that there is this data type. For example, if there's a pixel that has a value of 35, and now during some processing step, you try to subtract a value of 50, this will basically not work, and it will just give you 0. And the big problem here is that I think all softwares just do that without giving you a warning. So there can really bad mistakes happen if you're not exactly sure what you're doing. Similarly, if you try to add here too much, it will also not work and you will just get a wrong result. And so basically 255 is the highest value that you can get. Another thing is here, this decimal point is also ignored because the 8-bit cannot have decimal points. It's just integer values. Another very popular data type is unsigned short or 16-bit images. So here you can have higher values, but they only help you a bit. You still cannot have negative numbers. Now you can go beyond 255, but still the .2 will be ignored because it cannot be represented in this data type. Then another popular data type is float. So there you can have really, really big numbers, but this is not so much usually why this is useful. The really useful thing is you can now have negative numbers, which is good, and also you can represent decimal points. So this gives you here the correct result, 260.2. But in fact, also float is not perfect. It still has its own problems. So for example, if you have really big numbers, you get trouble. So for example, if you do this addition on the left, you get a slightly wrong result. So also here, there's some expert knowledge needed to do the proper things. So what you typically have to do during image analysis is you sometimes have to actually convert between these data types, because maybe you start with an 8-bit image as it comes from your microscope, but you need negative numbers because you do a background subtraction in a noisy image, for example. So you need a conversion. And I think here the rule of thumb is if you go from a lower bit depth to a higher one, it usually works. You should still check it, but I think typically the pixel values don't change, whereas when you go from a higher to a lower bit depth, you typically change your data. And again, most softwares will give no warnings, no errors. They will just do rather random things and you might not notice. So this happens, unfortunately, quite a bit. So you have to be really careful there. Okay, so after this very technical beginning, let's talk a bit about image formation. So again, if you interpret the gray values in your image, you have to also know two other things that can kind of change the values in some way, and that is basically things that happen during acquisition. So this is blur and noise. So here is an image of a cell, and I was drawing a line profile. So here on the x-axis, you see the coordinates of the pixels, and on the y-axis, you see the pixel values. And here at the edge of the cell, you see that this intensity peak has a certain width. And this width, if we look here at the x-axis, is maybe few hundred nanometers. And that's typically when you should be suspicious, because few hundred nanometers is the size of the wavelength of the light that we use for imaging. So that means that this width is typically not biological, but it's just a diffraction limit. So one has to know that one shouldn't interpret this width in a biological way. Another thing is you see here all these fine variations in the intensity of this protein in the membrane, and they could be real, but they could be also noise, because you have statistics when you basically collect these photons. Also your detector is not perfect, so you can't be really sure if this is real variation or a noise. To be sure, you would have to image the cell maybe several times and check if this pattern stays the same. Another thing one has to pay attention to when interpreting images is that this blur that I was talking about, it's typically anisotropic. So you blur typically more along the z-axis of your microscope system than along the xy axis. So I was drawing here, like the white should be the outline of a cell, so a cell membrane. And here I try to depict the small volume from which the microscope is collecting the signal, and which is anisotropic typically, as mentioned. And there also things can happen that might not be entirely obvious. So for example, if you look back at the image of the cell, you see at the edge of the cell, the signal looks brighter than at the center. So now if you're not very experienced, you might conclude that this protein is maybe specifically or more enriched at the edge of the cell. But if you know a bit about microscopy, you actually realize that because of the anisotropy of your point spread function, there's just more membrane fitting into the little volume that you image here at the edge than at the center. So this increase in intensity could be just an artifact of your imaging. So this is another thing. If you do bioimage analysis, you have to be really very careful to know which microscopy technique was used, what was the resolution, and so on, not to interpret your data wrongly. Now, since there's all these problems like noise and blur, there's in fact also a whole field, a subdiscipline of image analysis. It's called image restoration, and they basically try to develop algorithms that counteract these problems. So here's an example. You see actually both problems. We see this elongated, distorted shapes, which is because of the anisotropy of the point spread function that I was talking about, and it's also noisy. And in the lower image, it looks much better. So an algorithm has been applied both to denoise and to deblur. So traditional algorithms for denoising are probably just averaging filters. So I will talk about that in a second. And deblurring, you might have heard of deconvolution. So that's the classical thing that people do. In fact, the example I was showing here on the screen is not doing this. They use some more modern, now you have heard of all the deep learning. So they did some deep learning algorithms to actually in one go denoise and deblur to get in this case, I think, good looking result. As I said, an average filter is the thing you can do if you want to get rid of some noise in the image. So I would like to explain that a bit. So an averaging filter is actually a specific type of a so-called convolutional neighborhood filter. And the idea of all these neighborhood filters is the following. So if you look at this noisy image and you look at this little red square, it's a 5 by 5 pixel square. And there's one pixel in the middle, which is painted red here. So always when you do convolution filters, the recipe is actually rather straightforward. So you take all these values, in this case 5 by 5, so 25, and compute some mathematical function of them, which one then determines what filter that is, and you replace the pixel in the middle by this mathematical function. And then if you want to compute, for example, what is the value of this pixel next to the one in the middle, the 69, you shift this window of 5 by 5 pixel and you again compute this mathematical function. So I have done that here, and then you see how all these values are now much more even, and that's simply because we average a rather big local neighborhood, and that's why the noise also disappears. If you wanted to have it a bit more in detail, actually one important word here to learn is the notion of a so-called kernel. So here, as I said, we did a 5 by 5 neighborhood, and that is represented here. So we did a convolution with a kernel of a size 5 by 5. And then also all these 25 pixels got the same weight. And that's why they all have here a value of +1. And that's also, this is at the average, the square average kernel of a size 5 by 5. In fact, this is wrong, what we get here. So if you do that and you compute the average of all these values, the average is not exactly 54, but it is 50. Okay, I'm surprised now. Maybe I didn't take exactly the same data. It shouldn't be 52, but it's definitely a number with a floating point. Yeah, so it's not exactly an integer number. And this is one of these examples. Here, I didn't change the data type, so I went from 8-bit to 8-bit. So this averaging result is not exact, but it actually is rounded. So here again, already you have to decide, do I need these exact averages or not? And then I have to change the data type before. Just as another example, this is also a very famous convolution filter. It's a so-called edge detection filter. So here we have now a much more interesting filter. So we have +1s, 0s, and -1s. And the recipe is the following now. So if I want to get a new value here in the middle, I take the values on the left and I multiply all of them with +1. All of those get multiplied by 0, so they go away, and all of those ones by -1, and then I simply add all of this together and I get 116. And that's a rather high value, and that is because this pixel in fact sits on an edge. So in my new image, I have a high value. So this is another example, and you can imagine there's many things you can do, and it's also really a whole field of coming up with clever convolutional filter kernels. All right, so the aim of all this filtering typically is to make it easier to help to so-called an image. And what does segmentation mean? It means basically going from gray values to objects. Yeah, so that's what we quite often want. We want to, for example, count that in this image there are in fact 2 objects, and this is called a segmentation of the image. So how does that work? Again, this can become very complicated, but probably the simplest workflow is the following. So, for example, in fluorescence microscopy, it's often relatively easy because the objects are brighter than the background. So, and then the idea is you first do something called the threshold. So you go from your grayscale image to a so-called binary image, which can now only have 2 values, 0 or 1. And you basically say all the pixels that are brighter, I think in this case I was choosing 73, they should be above the threshold, so they are so-called foreground pixels with a value of 1, and if they are below the threshold, they are background pixels and they have a value of 0. So that's already quite nice. So we have now a background foreground distinction, but in fact we have not yet an object logic in this image. That's why you have to do one more step, and that's a so-called connected component analysis. And that is basically you look which of these foreground pixels are connected, so are next to each other, and then all the ones that are connected get the same value. And the result here is a so-called label mask image. So in this case, for example, all these pixels of the object on the right have a value of 2. And so the computer knows that all these pixels belong to object number 2, and the one in the left, they all have a value of 1. So that's how computers internally store objects in a so-called label mask image. So you go from intensity image to binary image to label mask image. Now, as I said, sometimes you need to filter before you can do that. And I just take the same data again. If we directly try now this threshold idea on our noisy image, you see we have a problem, and sometimes noisy pixels outside of the cells are actually as bright as some pixels inside, or pixels inside the cell are quite dim, so we miss them. So we get this problematic result here. But as we already have seen, we can be more clever. So we first do an average filter to get rid of the noise, and then we can easily threshold, and then we could do the connected component analysis. Now, finding good ways of making images such that you can easily threshold them is actually one of the core things that bioimage analysts are concerned with. So here is another very typical example in biology. So you have all these spots, in fact it's autophagosomes, and you want to segment them. Now you have the problem, if you want to threshold this image, it really doesn't work at all, because the dots in the nucleus, they basically sit on top of the rather bright background fluorescence of the nucleus. So in fact the whole nucleus is often brighter than spots that are in the cytoplasm. So here also an average filter doesn't help you. You have to do something else, and I want to introduce a bit more complex filter, but it's a very nice one. And it's a class of so-called rank filters. So that's a bit different from convolutional filters, but also not so different. So the idea is here that the first step is I look for each pixel, what is the dimmest value or the minimum intensity that I find in a certain neighborhood. That's why it's called a rank filter. Yeah? So you sort the pixel values and you take either the maximum, the minimum, or the median quite often. So I do that, and then what happens is that all the bright spots disappear, because for all these bright spots I can find a dark pixel in the neighborhood. What, however, also happens is actually this boundary, like the nucleus, will shrink a little bit, because each boundary pixel at the nucleus, for that one I will find a dimmer pixel in the cytoplasm. So that's in fact something that I don't want. So then the clever thing here is you take this first image and you apply the opposite operation, a so-called maximum filter. So I ask for each pixel what is the maximum that I find in a certain neighborhood, and then these pixels here at the boundary of the nucleus, in fact, they will become bright again because I find this bright pixel in the nucleus. However, these spots, they cannot come back because the intensities don't exist anymore in the image. So what happened is, in fact, we generated here an image that is a very good approximation of this kind of background of not locally bright signal. And now we can subtract this image from the original image, and we very nicely only get the spots, and we can easily threshold them. So it's a bit complex, but I thought I really show it because it also shows you can get quite creative here and have good ideas of how to basically treat your images that you can segment your objects. And this top hat filter is a very typical filter that's used quite often in biology. Now, this is all nice. When it comes to segmentation of electron microscopy data, then it's much harder, because in fact now the things that we want to see are not necessarily brighter than the things that we don't want to see, but it's really just more about finding different textures. So for example, here's the endoplasmic reticulum, which you can see with these two lines. Then cytoplasm is also quite complex, so it's full of ribosomes. Then the mitochondria, they again look somehow different, and then we have this other region here where I actually couldn't really find out what it is. Let's say we want to segment these 4 regions. That's pretty tough, yeah. So we would have, for all of them, find out some sequence of image filters that make them bright, such that we can apply our threshold. So we have to come up with this processing strategy for the mitochondria, such that we can threshold them, for the cytoplasm, for this other class, and for the ER. And that's really, really hard. I think it's maybe possible, but you need a lot of time and a lot of experience, and it's a really tough problem. But as you might have heard, machine learning is becoming quite popular in image segmentation. So the idea is you let the computer do all the work for you, and you help the computer by annotations. So what you do is you just paint in a few regions in the image examples, like say this is ER, here I have this other class, here's cytoplasm, here are mitochondria, so the computer knows that. And then what the computer will do, it will compute many, many, many different filters, like the ones we learned, for example, the edge, the average, maybe a top hat, many of them, and then automatically tries for you these filters or even combinations of these filters to see how to arrange them and what thresholds to use to reproduce your annotation. So the computer is basically doing all that, trying out different filters for you. And in terms of terminology, so the traditional filters that have been used for or approaches here have been that you just do one set of filters and then you compute thresholds on them. Now the more powerful approach that came around in the recent years, it's called deep convolution, and here the difference is that you don't, so you compute filters, but then on the filters you compute again filters. So you compute filters of filters, and you also usually downsample your image while you do that to get more context information, and that turned out to be a much more powerful concept. And this is now this deep convolutional neural networks that are at the moment state of the art for image segmentation. Okay, so once you have your objects, now you can actually measure something. And so I want to talk a little bit about what you could measure. And I think one can broadly distinguish 2 classes of measurements. There are shape measurements and there are intensity measurements. So for these 3 objects, for example, probably the first thing you would measure is how big are they. So you can measure the area. and you already see that's quite interesting because this cell, which is in fact in metaphase, so it's just dividing, has a smaller area because the DNA is condensed. So you can already use that, it's called a feature, object feature, to do some biological statements. Another example I put here is so-called circularity. This can be computed by taking the area and dividing it by the perimeter squared. And if you work that out here, it's actually some fun. If you do some school mathematics, you realize that this value is 1 if it's a perfect circle. So that means the closer this value is to 1, the more circular it is. And again, you see this is quite nice here. So for example, the interphase cells are more circular than the metaphase cell. And of course, there are many, many more of these shape features. I just gave now 2 as an example. You can also measure intensities. So for example, you can ask which of these pixels here in this object is the brightest. So this would be the maximum. And again, it's quite informative. So the DNA is most condensed in the metaphase. So that's why it has the brightest value. So the maximum is actually a good biological descriptor for this example. Then another thing you could do is you could ask what is the sum intensity. So you basically take all these pixels in one object and add up all of them. And also this is quite interesting here because it's basically not very different. And the reason simply is that, for example, here we have a pro-metaphase cell and a metaphase cell. So there's not much DNA produced if you go from pro-metaphase to metaphase. So the total amount of DNA is actually not changing. And that you can nicely see here that the sum intensity is actually the same. I also put this example because I quite often see publications where people just say we measured the intensity. And as you can see here, whether you measure maximum, sum, mean, median, it can be like extremely different. So this is a very important choice, which exact intensity you measure. So one has to be very specific there. And so just saying measuring the intensity is definitely not enough here. Now, you can try to, or you can be a bit more ambitious and try to calibrate all these measurements. So in fact, if I show here the column headers again, so we measured the area in pixels. Circularity is a bit special. So we divided, in fact, 2 values in a way that the unit canceled out. So these are measurements that don't depend on any calibration, and that's actually quite nice. So these are often good values because they are quite robust. Then the intensities, they were both in gray values. Now, what we could, for example, do is to measure the area not in pixels, but in micrometers squared. And that is usually rather easy to get because you typically know how big each pixel is in your image. You just get this from the metadata of your image, which is stored at the microscope. As said, for circularity, we in fact don't need it. Now for intensity, it's much more tricky. Yeah? So what would be really nice if we wouldn't say intensity in gray values, but in concentration. So for example, number of molecules per micrometer cube, or some intensity would be then total amount of molecules in a certain region, in the whole objects, for example. So this is in fact also possible. And I have put here a paper where you can look. And what they did, they used a technology called fluorescence correlation spectroscopy, which allows you to determine calibration factors to go from gray values to concentration. So if you can do that, that's of course very nice because you get real biophysical measurements rather than just gray values. If you do bioimage analysis, quite often what you do is you have many images and then you have later many numbers, which then typically you go on in Excel or R or Python, MATLAB to basically do the statistics, which is fine. But if you have all these numbers in these tables, there might be some outliers or very high or very low numbers. And you should be curious as a scientist, is there this high number in this column? That basically means you want to actually see how did the object in the image look like that produced this high number. And that can be actually a bit tough because if you have this table open in Excel, how do you find back the cell that this number belongs to? And so I call this step results exploration and quality control. And that's very important. I think it's essential. And so you kind of need to think about what software, for example, you can use to make your life a bit easier here. So actually, I think what one should look for is a software like this. So let's say you make this scatter plot. So in this case, each dot is a cell. And actually, the value is how many Golgi fragments were segmented in that cell. And different treatments here give different values. And then you see, okay, if we treat the cell with nocodazole, there's really a lot of Golgi fragments. So naturally, I think you want to know, how does the cell look like? And also, is it true, or was maybe just my algorithm that segmented the Golgi not working? Yeah, was there maybe just noise? So this is the questions you have to answer. So I think what you really want is a software where you can just click on that data point and it opens the image for you. It tells you, maybe put a cross on it, which cell it looked at, and not only shows the raw data, but also here in colors the segmentation of the computer. So where the computer thought the nucleus was in blue, where the cell outlines were, and where the dots are. I think this is really essential to have that kind of workflow easy. And also then you could click on the control cell and you see, aha, yeah, the Golgi indeed is together. So here you then are happy because you see it all worked perfectly. I really just had a very strong phenotype. So I think this is essential to do some quality control like this. Another thing that one should do is you should manually annotate a few of these cells where you basically manually try to find, for example, the Golgi in that case, and compare this to what the computer did. And then you can compute different values. Two standard ones, I think, are called precision and recall. So in precision, you take the true positives, so the number of things that have been correctly identified by the computer, and divided by the number of true positives plus false positives. And the recall is a similar number, so I think I will not go into detail. I actually just put a link here on Wikipedia where you can read about this, but this is basically also, I think, becoming more required, that you characterize your automated analysis in such a way. I want to just say a few words, because now I think there's quite often is also talked about image ethics, and I actually also already gave a workshop about it. And it's about what you are actually allowed to do with these images, yeah? How you can treat them or not in your image processing. And it's a good discussion, but it's also interesting that it arises, because in fact, I haven't heard yet somebody asking about microscopy ethics. But there also, imagine you sit in front of the microscope, and I mean, you will often decide which cell you actually image, and then you will not image other cells. You might decide in which focal plane you image, and you might decide the pixel size if it's a confocal. So if you make it bigger, you will not see small structures. You will also decide the detector gain. If you put that very low, you will not see dim structures. So actually on the microscope, there's tons of way to manipulate in a way, or tweak, I don't know, what you will see in the end or not. So I think also there a discussion of ethics would be very appropriate, but somehow I think it's not happening so much. And I think maybe the reason is that if you go to a microscope, you already feel like a scientist. Yeah, so you're doing already your science, you're in a science mode. And maybe it's just a hypothesis. The thing is that if people see images, maybe it's more that you also think about daily life images, and then maybe Photoshop, and then maybe it can easier happen that you apply the image, let's say, processing that you would apply in daily life to scientific images. And that's why this image ethics discussion is maybe arising. So I think the point is basically just to realize that this scientific bioimage analysis is very different from what you can do in Photoshop. And it's really, as I said, a professional thing where you maybe need a bioimage analyst also for consultancy. Now, in terms of what you can actually use to do bioimage analysis, I try to make here a list. And the first thing you see that the slide is rather full and has both good and bad. So the good thing about is that you can see in basically all major programming languages, you have something. So you will for sure find a solution to your problem. The kind of bad news is that it's very difficult to actually know which of them to use. And or even for myself, I'm now quite for many years in this business, find this very confusing to choose any of these tools. And that is, I think, really just how it is. I don't have a good answer here. What I maybe is a bit of an answer, and I put it a bit outside, is a software called KNIME, where basically you can generate workflows, also image analysis workflows, but in fact whole data analysis workflows, combining tools from, I think, in fact, almost all of the languages that you see above. So maybe tools like that might be the answer to this complexity. With this, I would like to thank a lot of people here that provided data and also input to the slides. And then I would like to thank you for your attention, and I'm happy to take questions.
Thank you very much, Christian. So if you have any questions, please put them in the question section of the GoToWebinar panel. To get us started, we have some questions ready. This following question was submitted in advance of the webinar, which is, would you recommend scientists doing bioimage analysis on their own or seek professional consultancy?
Yeah, thank you for this question. As I said in the beginning, it's very complex task to do good bioimage analysis, because you have to know all these computational methods, but at the same time you really have to also know how exactly was the microscopy done, what is the exact biological question, how was the sample stained, was it an antibody, was it a GFP? So it's very multidisciplinary. So I think the ideal case is probably there is a professional bioimage analyst in every institute, and then people can, like the scientists, can discuss with this person what to do. I think that's probably the ideal setup.
Okay. Our second question here is, can you identify over and under saturation based on only the pixel intensity distribution without knowing detector sensitivity?
Yes, so I hope I understand the question properly. So I will go back here to the data type slide. So what I typically do, one of the first things if I get images that I should analyze is I look at, in fact, how, like if it's an 8-bit image, how many 255 gray values do I find in the image and how many zeros do I find. So if I find a lot of 255s, then this is a sign that it has been saturated, the detector. If there's a lot of regions with plain zeros, that's actually a sign that it has been undersaturated. So basically the signal was cut at some point, which is, it looks nice because you have a very clean background, but in fact it's not so good because you don't really know where your zero level was. So this is also not a good image. So I think, yes, by just looking at how many values you have at the extreme of your bit depth is a way to check this image.
Okay,
yeah.
Good.
Mm-hmm.
Just a question related to that slide. There's a question, why is 255 assigned as 2 raised to the power of 8 minus 1?
Can you repeat the question?
Why is 255 assigned as 2 raised to the power 8 minus 1?
So I mean, first of all, it is just, if I didn't make a mistake, I hope it's just what happens. But
yeah.
But the reason is basically so if you have an 8-bit image, how this is stored in the computer is a sequence. It's basically 8 numbers, and they can be either 0 or 1. And depending on where the 1 is in the sequence of these numbers, it's interpreted as 2 to the power of 2, 3, 4, 5, 6, 7, 8, and then you basically add up all of those. So I think I would need to, I can't explain it without drawing, but if you just Google for how computers represent an unsigned byte, then I think you will see how that works. Yeah.
Okay. Our next question is, for image analysis, should we convert a 16-bit image to 8-bit, or should we not do it because it loses depth?
I think you should not do it because it loses, in fact, it loses not only depth, but the problem is if you go from 16 to 8-bit, you also have to make some decisions because, for example, in your 16-bit image, you might have a value of 3,500, but you need to have something that's 255 or lower. So there has to be some mathematical formula. If you really need to go to 8-bit, it's possible, but then you have to basically really think about that formula and apply the same formula to all images that have been acquired on the same microscope and with the same settings. So it's not strictly forbidden, but it's very risky and one can make a mistake here. So I would at all costs avoid that if it's not really, really needed.
Okay. Our next question is, do you think it makes sense for a biologist to invest time in learning all these analysis methods or just collaborate with someone?
Yeah, that's a bit like the same question that we had in the beginning.
Yeah.
It's I think, I mean, I don't know. I think many of the, or some, or a good fraction of the now, let's say, more people that think of themselves as professional bioimage analysts, they started off as biologists. So it's not that only computer scientists or physicists become bioimage analysts. So if you feel like you find that interesting and you like mathematics and you like computers, I think you can totally go for it, and it can even become a career path for you that you become good at that.
Okay. Our next question is, could you please point to some practical examples where NIME was solving some common image analysis issues?
So I don't have a specific project, but for example, what I can say is I just actually this week, Monday, Tuesday, Wednesday, I was running a course here at EMBL about basics of bioimage analysis. So the first 2 days I was showing things in ImageJ. And then on the third day, we basically repeated the same concepts in NIME, and the response was very good. So people really, really liked it. They wanted to have a follow-up course. So I think it's as I said, you can do this in any software. You can also write C code, of course, but I think it's very visual, and I think people usually pick it up rather quickly. So, but yeah, you can, as I said, unfortunately, there's a lot of choices, and I don't think NIME is the only one. Yeah.
Okay. On the slide with the derived measurements, you listed total intensity with 6 significant digits. Does the accuracy of the measurement warrant such precision?
Wow, yeah, that's a good question. My gut feeling is no, but I would, to be honest, in this specific case, I would have to sit down and think and write some formulas. What really I think is the precision here. So it's a very good question, and I probably not, but I cannot give a mathematically clean answer now what the good precision would be here. But very well spotted. So yeah.
Okay. How many images should I analyze per one data point on a plot to be sure my analysis was thorough?
The answer is until your t-test becomes significant. So you can't know before. Basically, what you want is to have a statistical significance. And if I look, so can you actually see my mouse pointer here?
Yes.
Yes, okay. So to reach statistical significance, there are two things that play a role. One is the difference between the average values. Yeah, so if the difference between the average values is very high, then you quicker reach statistical significance. And the other thing is if the spread, so the sigma, if that's very low, you also quicker reach statistical significance. So basically how many images you have to take is a function of the difference between the means and of these two sigmas. So you can't actually know how many you have to take before you didn't make this plot at least once.
Okay. The next question is, what is the best colony counting program for non-round cells?
I don't know.
Okay.
So, I mean, for example, if let's say in general, if you want to have a solution where you don't need to know too much, and it's more this machine learning based approach where you basically draw annotations, I think the most popular software is something called Elastic. So ILSTA Okay, maybe I misspelled it. So elastic, and that's very popular for general segmentation purposes. So that's the maybe one I can name.
Okay. Could you elaborate more about KNIME? Is it user-friendly or some basic programming is required?
Unfortunately, I don't have a screenshot. It is user-friendly, so I think if some people have used LabVIEW, maybe not. So you don't have to write any code. It's in the end the workflow is little graphical icons on your screen, and you go from one step to the next with a little cable between them. So it's really it's very visual, so no programming.
Okay. The next question is, where does image registration fall in your workflow? It is usually used to get comparable data in medical diagnosis, or is it ever used at the biomolecular/cellular level?
So I actually I work a lot on image registration, maybe one of the main things, in fact, since recently. So it's typically maybe even the first step of your pipeline. So I mean, for example, we have here a screening project where people look at Drosophila, and it's 3D data. So they have thousands of 3D images of Drosophila, and they lie in kind of random orientations on the microscope slide. So really the first step is to get all of them in the same 3D orientation. So there's registration. I think yes, it's a big thing also in biology, but it's also interesting that you mentioned medical, because in fact I was looking around for what's in this case the best software to do that, and in fact it comes from the medical community, I think, and it's unfortunately a very similar name to the software before. It's called Elastix. So not ilastic, but Elastix, and that comes from the medical, but I use that a lot now for also biological data.
Okay. Our next question is, how much knowledge in programming is it required to be able to do basic bioanalysis?
I think 0, maybe. So for example, there's also commercial softwares like Imaris. They are really user-friendly, and also in ImageJ you can do a lot. I think it's 0. It becomes a bit more typically if you want to do batch analysis. So if you want to do the same thing several times and basically loop over a folder, then depending on the software, you sometimes need to write a little bit of code. But I think also KNIME is an example where you can get very far without any programming. So I think you can get quite far without.
Okay, so it doesn't look like there's a follow-up question.
Okay.
And I think we're almost down to our time now. So if that's all the questions we have today, I would like to thank Christian again for this interesting webinar. As mentioned earlier, our webinars are recorded, and these will be made available on the CORBEL website and the YouTube channel in a couple of days under the webinars page. Christian, if you could go to the end slide.
Yes.
Perfect. So you can also check out our upcoming webinars on the webinar page of the CORBEL website, and also be updated through our CORBEL newsletter. And all information can be accessed through the URL as seen on the slide here. Again, thank you everyone for attending, and thank you, Christian, again, and we will see you in the future webinars. Thank you. Goodbye.
Thank you.